starting with the next chapter, and that is immunity. Now, when we're talking about immunology, this is a chapter which will be very important in a lot of subjects, starting from pathology, microbiology, medicine, biochemistry. So there are a lot of overlapping topics that you all are going to get over here. So let's begin first by talking about an introductory portion so that we get a hang of all the important cells, their markers, their functioning, and then we can go on to the other set of disorders. So let's first begin with what are the types of immunity that God has given us or what we have acquired on our own. So what did I say? A type of immunity that God has given us, that is one type, or something that we have made of our own, so that is the other type. So the one that is a God gift, that is referred to as the native or the naive or the natural or the innate immunity. These are all the names that are given to it. You can see all the N's coming up your way. So repeating once again, that is known as the native or the naive or the natural or the innate immunity. All these N and N's are God-gifted ones. Okay. So this is the one that is present since birth, as God had gifted it. On the other hand, if they talk about something known as acquired immunity, means we have acquired it or we have adapted it, that is known as adaptive immunity. And this immunity is the one that we have acquired after infection, after exposure to some particular bacteria, virus, parasite, fungus, and so on. So simple, there's one God's gift which we have since birth, that is innate immunity. There's second immunity which we have acquired after getting infected, that is acquired or adaptive immunity. Which out of these do you think is going to have features of memory of some bacteria or virus? Of course, this is what happened after exposure. So this will have a memory. And which of them is going to have a specificity for some bacteria or a virus? Again, the acquired one. This means natural immunity neither has any memory because God has given it just as a gift. No memory of any bacteria, virus. Neither does it have any specificity against any bacteria, virus. It's just a simple way of learning. It's a God's gift. Now you need to know what are all the things that God has gifted you. What are all the components? What all things in your body come under the natural immunity? Let's make a list. So starting first with what are the innate immunity components that you need to know. So first, think logically, epithelial barriers. Don't you think that God has given you skin and God has given you mucosa, and these are nothing but your epithelial barriers? Like if a bacteria wants to come in through your skin, it's going to have a problem. If a bacteria wants to come in through your respiratory mucosa, your GI mucosa, so these are barriers which God has given you. So first one is epithelial barriers. What is the second one? Cells. God has given you certain cells, like for example neutrophils and macrophages. What do neutrophils and macrophages help in? They help in eating up, phagocytosis. So they will kill, this is again, God has gifted you with these cells in your bone marrow. So they will kill the bacteria, viruses for you, and they will help you in phagocytosis. They will help you in immunity. Just a word of caution over here. When I'm talking about macrophages are going to be a part of innate as well as acquired immunity, which you will gather in a while. Macrophages are going to be a part of innate as well as acquired immunity. This could be a question also. Moving on to the next. The next we have, what we have is the complement system. So in the complement system, it is the alternate complement system that God has given you since birth. And why is that so? Because alternate complement system is the one that is activated by IgA, and IgA is again an antibody which is present in all of us. It's a mucosal antibody. It's present in all the mucosal lining. So alternate complement system is innate immunity. By now, these three things you know. You have studied epithelial barriers. It's, you know, body is made up of skin and mucosa. You studied phagocytosis with neutrophils and macrophages. Now what you need to know, third one, complement pathway. Have you studied that also in your, in general path, in general micro? You have studied all these complement systems everywhere. Now what we need to know is these two cells. The first one is the innate lymphoid cells or NK cells, and the second is PRR, that is pattern recognition receptors. So let's begin one by one. Let me start with the innate lymphoid cells or the NK cells. What are all the names? Very, very important. When I'm talking about NK cells, which we call as the natural killer cells, what are all the names of the natural killer cells that we have to know? First and foremost, they are innate. Every word will make sense. You don't have to mug up anything. Are they innate? Yes, they are innate. They are a part of innate immunity, God-gifted immunity. Are they lymphoid cells? Yes. So please note how many types of lymphocytes has God decided that your body will have? God said that you will have either B lymphocyte, or you will have T lymphocyte, or you will have a third lymphocyte called as natural killer cells. So are they a kind of lymphoid cells only, like I said? Yes. So simply repeating, this is an innate lymphoid cell, or ILC. But are they B lymphocytes and T lymphocytes? No. B lymphocyte and T lymphocyte is totally separate. NK cell is totally separate. So can I say NK cell is nothing to do with B and nothing to do with T? It is a non-B, non-T cell, means it is a null cell. It has no B cell, no T cell properties, non-B, non-T cell. Okay, so then you will ask me that what are the structure looking like? So firstly, it is not going to be under the control. Usually, this is a concept which you might not understand right now, but gradually you will analyze this and understand that whenever I want to get an antigen to my cells, to my B cells or to my T cells, I want to give a bacteria, a virus, I have to bring it with MHC. MHC brings a bacteria or a virus for me. NK cell says, I don't need any MHC to bring any bacteria virus for me. I am God's gift. I have God's blessings. I am going to find out the antigen on my own, bacteria virus on my own, and I'm going to kill it. I don't need that any MHC should come and give it to me. So this says that I have nothing to do with MHC. I am an MHC unrestricted cell. Next, what does it look like? Are these lymphocytes? Yeah, they're not B, non-T, but lymphocytes to hai. So I'll draw a lymphocyte over here, but I've drawn a huge lymphocyte because overall the shape of this is very, very large. It's a large lymphocyte, and I'll draw this as the nucleus, and it's also got a lot of granules. So remember, it is an LGL, large granular lymphocyte. That's another name given to it. Can you imagine? All these are the names of the NK cells. Natural killer cells, they are a part of innate immunity, so they are innate lymphoid cells. They are lymphoid cells, but they are not B and T, so they are called as non-B, non-T cells or null cells. They don't need any MHC to bring an antigen to them, so MHC unrestricted cell, and their structure is LGL, large granular lymphocyte. But right now, you said that they have no B cell properties, no T cell properties. So this means all the markers, CD markers of B cells and T cells are also not going to be there. That is why I call them null cells. So immediately you will ask me, what are the NK cell markers that you have to know? So please remember 16 and 56. 16 and 56. And if you go by how the bodybuilders take measurements, and they're very fond of numbers as such rather than fitness. They're very, very fond of numbers. So a 16-inch biceps, and we all know the favorite meme that was created on the 56-inch chest, right? So remember 16 and 56. These are the natural killers. Someone has 16-inch bicep and a 56-inch chest, that person is going to anyway kill anyone. So 16 and 56. But now the recent INI CET question was, is there any new marker? And yes, there is something called CD94. I will be telling you the meaning of all of these in a while. You have a person who has a 16 and a 56, CD16, CD56, and a new marker that is CD94. So now I know that they are totally different. They have nothing to do with B and T cells. They are totally different set of cells that you have. Let's do their function first. Please note, natural killer cell. Whom do you want to kill? If I ask you as a, forget natural killer cells, if I gave you the responsibility of killing, whom would you want to kill? Would you kill your own cells or will you kill a foreign cell? Of course, you will always and always target a foreign cell. You will never ever kill the own cell of your body. That is never going to happen, right? So please analyze over here that NK cells, if this is an NK cell, for example, every NK cell has been given a start and a stop button. Means there is one arm that God has given it, which is known as the activating arm. This is known as the activating arm, and activating arm is going to kill. It is the God's gift to kill. So I call it NK God. NK God matlab NK G2D arm. NK matlab natural killer. G2D I've learnt as God because the God's arm that has given to NK cell to kill the bacteria. So NK G2D arm is the activating arm. And then there is a stop arm that has been given. That is an inhibitory arm. And do you know inhibitory arm is CD94? That CD94 new marker that I was telling you, that is nothing but the inhibitory arm. Okay, so moving forward, if for example this is a natural killer cell, one start button, one stop button. If I now give it a self cell of your body, a self antigen, say your red blood cells, your own heart antigens, your own joint antigens, all of our own, if I give it anything of my own, is it going to kill it? No, I can't kill my own self. How? Because every cell of my body expresses MHC1. So natural killer cells have decided that whenever I will see MHC1, it's like an ID card. It's like MHC1 I hai na, to ID card hai. Like if I ask you what is your name, you will show me your ID card, your Aadhaar card, your ID card, and show me your name in it, right? So then I'll get to know that, okay, yeah, you are my student. I recognize you. Similarly, if I want to recognize my own cells of the body, God has given me an ID card that is MHC1. So as soon as MHC1 will be seen, natural killer will say, this is my own cell. I don't have to kill it. So which arm is it going to put forward? Stop wala arm. That don't kill it. This is our own student. This is our own cell. So remember that whenever I see MHC1, means it's my own cell, which is the arm that I'm going to put forward? Inhibitory arm. I wala concept yaad rakhna. So remember, MHC1 is seen. This means the inhibitory arm means you're not going to kill your own cells. But now let me draw the same NK cell. Let me draw an inhibitory arm. Let me draw the same activating NKG2D arm. This time, the same self cell of your body has now something different. It has been infected by viruses or it has some cancer growth happening. Now, do you want to kill this cell? Ye to simple cell nahi hai na. This is a cell which has a virus or a cancer. Of course, I want to kill it. But how will I kill it? Because viruses and cancers have got their suicide mechanism with them. Viruses and cancers are going to decrease the expression of MHC1. That is what they do. Viruses and cancers, they decrease the expression of MHC1. So ID card gaya. Now the ID card is gone. Now I'm not going to identify it as my own cell. Now which arm is going to come and destroy? This activating arm is going to come and is going to kill whatever cell is there. So simple. If I see MHC1, it is my own cell. It is I, myself, and I will put up the inhibitory arm. So MHC I dikha, 1 dikha, I myself hai, wo self hai, and that is inhibitory arm. Whereas if I don't see MHC1, why will I not see? Because viruses and cancers decrease the expression of MHC1. So which arm will I put forward? We are going to put forward the activating arm. Everyone's understood this. So NK cells are doing a very nice job. They've got start and stop mechanism. They're doing very nice job. But who is making them do this job? This job is being done by certain interleukins. Remember, interleukin 2, 12 and 15. If you are asked what are the interleukins of NK cells, they are 2, 12 and 15. What do you have to know about them? Someone asks you, how do you stimulate them? Listen, do some. First, I'll have to push you a little. Thoda pad lo, little bit you should study. It's good for you. So for stimulation, you have 2 and 15. For stimulation, you have 2 and 15. After you start, if you get stimulated, after you start studying, then I'll have to tell you, acha, jab pad liya hai, thoda MCQ bhi kar lo. I will tell you to kill the final target. Kill the final target is not reading theory. Killing the final target is doing your MCQ. So who is going to do that MCQ part of it? Who is going to help in the final killing? 12 is going to help in the final killing. This is just a story to make you learn. First, you learn the 3 interleukins, interleukin 2, 12 and 15. After that, you say, first you have to stimulate. Like I said, I will motivate you that please thoda sa pad lo, study a little bit. So how will I motivate you? By interleukin 2 and 15, I will motivate you. After you are motivated, after you started doing a theory, now I'll say kill the target, kill the MCQ. How will you kill the target? That is going to be by interleukin 12. Okay, good story. 2, 12, 15 will you be able to remember? Revising the CD markers once again with you, CD 16 and 56, and the new marker. Now you know what is this new marker? The new arm marker, that is CD 94, is the inhibitory arm that we have. So this is sorted. Now guys, please listen to me very carefully. There is a process known as ADCC. What is ADCC? It is antibody dependent cellular cytotoxicity. Antibody dependent cellular cytotoxicity. And what do I mean by this concept? Please note that whatever cell I have, whatever cell, maybe it's infected with some virus or maybe it's some cancer or any other infection that I'm talking about. You know, what does it get coated by? Everyone is aware it gets coated by something known as opsonin. What is the opsonin? The opsonin is IgG. So it basically gets coated by IgG. So my concept is that if I want to kill this virus, I will have to pick up IgG. who is going to be picking up IgG? So neutrophils, macrophages, eosinophils, even NK cells. These are all the ones which have a property that we are going to go and identify. They are going to go and identify this IgG. So actually, instead of writing it like a round, I think you should be able to pick up IgG like this. This is what you have as IgG, right? So this is what you have as IgG. That is how it has gone and gotten attached onto the virus. This is IgG. So what will you do? Which part of IgG should you be able to target? Now these cells, neutrophils, macrophage, eosinophils, NK cells, they should be able to go, like for example NK cell, they should be able to go and attach to the Fc portion of IgG. This region, this last point is the Fc portion. So can I say that in NK cells you had written CD16 is the receptor for the Fc portion of IgG. Now you understand that line, that when an NK cell will go, for example, when an NK cell will go, how will it bind to the Fc portion of IgG? It is going to bind to it by CD16. CD16 binds to the Fc portion of IgG, and that is what is known as antibody dependent, because of an antibody it got attached onto it and then it is going to kill it. So it is antibody dependent cellular cytotoxicity, and not just NK cells. You will study later on in a lot of hypersensitivity reactions. Neutrophils, macrophages, eosinophils, NK cells, all of them can also do that. Repeating once again, this is some virus infection. First, I will make it tasty. How do I make my bacteria viruses tasty? By coating them with IgG. So now if I want to kill that virus, kill that bacteria, I will need IgG ka receptor. So I'm going to have a receptor for the Fc portion of IgG, which in case of NK cell is what? CD16. Go and attach, kill it. That is known as antibody dependent cellular cytotoxicity. That definitely wraps up my story of the NK cells. We were on innate immunity. Innate immunity mein NK cells are also done. Yes. I'm left with the last one. What are PRRs, pattern recognition receptor? Have you ever wondered, bacteria enters my body, virus enters my body, parasite, fungus, anything enters my body. How do I get to know that this time a bacteria has come or a virus has come or a parasite has come? How would I get to know? I will have some a secret agent who is telling me, giving me that information that listen, this time bacteria aaya hai. Listen, this time COVID has come. So who are those secret agents? They are known as PRRs. What do you understand by PRRs? Pattern recognition receptors, which basically means that I will have receptors in my body which will recognize the pattern that, okay, this is the pattern of a bacteria, this is the pattern of a virus, this is the pattern of a parasite. They are doing that for me. So this time I'm going to draw one cell over here, and in that cell I will say there are two receptors which are present on the membrane. Which are the two receptors which are membranous? The two receptors which are membranous are take care on the membrane, that is toll-like receptor and C-type lectin receptor. Toll-like receptor written as TLRs. Toll-like receptor and C-type lectin receptor, they are present on the membrane. And by membrane, I don't mean just the membrane of the cell. It doesn't mean only cellular membrane. Toll-like receptors are also present in the membrane of the organelles. Any kind of organelle like an endosome, this can also be present. For example, you have an endosome. So endosome ke bhi membrane par you can have toll-like receptor. The concept is some membrane they will stick onto. They won't be floating around. They'll stick onto some membrane. So T and C will stick to the membrane. Whereas whatever is left, that is, we have rig and nod-like receptors. So rig and nod-like receptors will be floating around. They will be cytoplasmic. Okay. They will be floating around. They will be cytoplasmic. So first I've analyzed there are four receptors that God has given me. Take care, that is toll-like and C-type are present on the membranes, whereas rig and nod are going to be roaming around in the cytoplasm. But what is their function? So remember, toll-like receptors are going to be for almost everything on this planet. But primarily we study them from point of view for bacteria. So whenever a bacteria, they are going to identify what? So please remember, toll-like receptor is going to identify a bacteria. But right now I'm going to tell you toll-like receptor is for a lot of things. C-type lectin receptor is going to be for fungal organisms. Rig-like receptors are going to be for viral organisms. And nod-like receptors are my favorite because nod-like receptors are going to identify dead cells, that is necrotic cells. It can also identify organisms, actually some bacteria, virus it can identify, and it can also be associated with certain diseases. Diseases kaunsa wala? It has been implicated in diabetes mellitus. It has been implicated, remember, NOD2 mutation kahan hota hai? Where in surgery, where in pathology have you studied NOD2 mutation? Inflammatory bowel disease. Inflammatory bowel disease also has NOD2 mutation. So let's do a very quick recap. When I'm talking about the NOD receptors, and I'm going to make you revise, if you guys have studied general path and the other chapters of general path, you know about it. What is NOD2 receptors? N for necrotic cell, O for killing the organisms, D for diseases. Diseases like D for diabetes and D for bowel disease. Bowel diseases may be iske saath it has been implicated. Please note, when I say that it is helping in the killing of organisms, do you think of some cell death? Very, very important cell death that I want all of you to know. Which was the cell death which was associated with NOD2 receptors or NOD receptors. Remember the cell death which was with bacteria, pyroptosis. Go back to your pathology chapters, go back to general path, go back to pyroptosis. Over there I've told you a question that in pyroptosis there is NOD2 receptors because NOD2 receptors pick up bacteria viruses, na? And pyro word means fever, so there was bacteria viruses over there and NOD2 receptor was helping me do that. So coming all the way back over here, a quick recap of this cluttered slide that we have made. There are 4 receptors: toll-like, C-type, Rig and nod. Toll-like and C-type, take care, are going to be present on the membrane. Other ones, that is Rig and nod, they will be floating around in the cytoplasm. Then toll-like receptor is mainly for bacteria. C-type lectin is present in fungus. Rig-like receptors are going to be present for viruses, and nod is going to be nod for necrotic cells, organisms and diseases. This is your basic. But now you need to know what are the different types of toll-like receptors. So when I'm talking about toll-like receptor, you can see a long list in front of you, and remember, toll-like receptor usually goes and works how? It tends to activate the NF-kappa B pathway. It tends to activate the NF-kappa B pathway, right? Now, there are lots of toll-like receptors as we can see. So, let me try and simplify it a little at least. So, when I talk about toll-like receptor type 2 versus toll-like receptor 4, I'm taking the even number first. Okay, so 2 and 4. So, 2 and 4 may I see that gram-positive bacteria it will identify and TB it will identify. How will I learn this? Have you read the cell wall structure in microbiology of a gram-positive cell wall? Does a gram-positive cell wall have something called teichoic acid? Yes, it has something called teichoic acid. So please remember, teichoic acid wala gram positive and TB, to TB or teichoic acid ke liye 2, TTT, TB and teichoic acid ke liye I need toll-like receptor 2. Is that okay with everyone? Easy to learn. Simply once again, for TB and for teichoic acid, I need TLR2. So gram-positive bacteria done. So 4 then becomes for gram-negative bacteria. So the bacteria story seems to be sorted. Let's move forward. What do we have with 5? Remember, F for 5, F for flagellin. If I have to identify flagellin, I'm going to take up F and F, F for 5, F for flagellin. It becomes so simple to learn, no confusion again. Okay, let's move forward. Now I have to focus on what? I have to focus on mycoplasma. Mycoplasma is what? Mycoplasma shows you which colony? It's going to show you fried egg colonies. It shows you fried egg colony. So eggs कैसे purchase करते हो? A dozen eggs, half a dozen eggs. So या तो dozen में होगा या half a dozen होगा. तो 6 goes well with half a dozen. So simple when we say या तो dozen या half a dozen. That is how you purchase eggs, na? So toll-like receptor 6 is going to be those 6 eggs that you have purchased from the market. So that is going to be the mycoplasma fried egg colony. अब सब हो गया. Bacteria is done. Now I'm going to come down to viruses. Viruses में I have simple. I have RNA viruses that I have to focus on, right? So when I say RNA, so most commonly I have single-stranded RNA. More common RNA is single-stranded. Single-stranded 7 के साथ है. So remember, when I say single-stranded RNA, it is associated with toll-like receptor 7. And uska almost half kara. 7 ka half is what? 7 ka half is 3.5. I can't have 3.5. So I'm definitely going to have 3. So the double-stranded is going to be with toll-like receptor 3. That is how I've learned the RNAs. Again, it's tough. You will need repeated revisions, but we'll work our way out. Repeating once again, when I'm talking about single-stranded RNA, single matlab 7, and double-stranded ke liye I do it half, that is going to be TLR3. What am I left with? If I have to identify the CpG, what is CpG? C and G, cytosine, guanine. If I have to identify CGs in a DNA, so the way you write CpG, from today onwards, P ka ulta karna, you will get the toll-like receptor that identifies CpG DNA, and that is TLR9. I'm going to revise entire table once again for all of us. When I'm talking about gram-positive, and gram-positive I know has teichoic acid in it, and then TB also has T in it, so 2 is the toll-like receptor that I will focus on. Easy one? Okay, when I'm talking about RNAs over here, single-stranded RNA is going to be single for 7, and double-stranded RNA uska half, that is going to be TLR3. When I talk about gram-negative, this I've learnt as it is going to be TLR4, because if positive was 2, तो अगला even number is 4. Now I come to flagellin. Flagellin is 5, so TLR is going to be F for flagellin, F for 5. Now we come to mycoplasma. I have gone to the market to buy half a dozen eggs, so that is going to be TLR6. And then when I write CpG, so CpG I ended up writing it only 9 instead of CpG, so that is going to be TLR9. As I said, this is a very, very tough table. No matter how many mnemonics I may give you, you will always need a revision of this. I hope you are sorted. There's one more thing that you have to know out of all of these coming back. So TLR ka extra point we have done. Now I'm going to tell you the extra point related to these RIG receptors that has come out. Regarding the RIG gene, you have to know something known as interferonopathy. So first you need to understand what is a RIG receptor. You don't have to know the full form. Let me tell you beforehand, you don't have to learn the full form. You just need to read through it once. It is retinoic acid inducible gene. So that by chance it is written as retinoic acid inducible gene, you should be able to put it together and analyze that, yeah, this is RIG. What does RIG receptor activate? RIG receptor activates STING. Again, STING, you just have to know that, okay, yeah, it is a stimulator, ST for stimulator of IN for interferon, G for gene. It is a stimulator of the interferon gene. So my basic concept is that when the RIG pathway will be activated, then the STING pathway will be activated. And if I'm saying interferon gene, remember which interferon is going to increase? Interferon alpha is going to increase. Interferon alpha increases, and that results in a group of diseases known as interferonopathies. That is known as interferonopathies. So having said that, what are the diseases now? Now they are doing a lot of studies on this. Now they're saying things like SLE are also being included in interferonopathies, where a lot of interferon alpha is increasing. So simple. RIG is retinoic acid inducible gene. Ab to yaad hi ho gaya. That is going to activate STING, which is a stimulator for the interferon gene. And which interferon am I increasing? That could be a question. So you should know these short forms, RIG and STING, and which interferon? Interferon alpha. On that note, we have managed to finish all of our innate immunity. The innate immunity components are done. If I go back, I have finished off the native or the innate immunity. Now I'm going to go on to the adaptive or the acquired immunity. And what all is going to come under adaptive or acquired immunity? It is of two types. One is going to be the cellular immunity. One is going to be the humoral immunity. Cellular immunity is going to have cells like the B lymphocytes and the T lymphocytes, and humoral immunity is going to have all the immunoglobulins. Let's begin. I have got you a table. First, I'm starting with cellular immunity, and under cellular immunity, I've got you a table of B and T cells, which is very, very important to differentiate. If I take a blood sample, if I take your blood sample, what is the most common cell percentage that I'll get? 60 to 70% of your cells are going to be the T cells. 60 to 70% of the cells are T cells. B cells, on the other hand, are lesser. They are only 20 to 30%. Still, it is not coming across as a full 100%. What if 70% are T cells and 20% are B cells? So, 100 to nahi bana. So, is there any other cell also which is almost less than 10%? What cell is it? Which is the third lymphocyte which I'm missing out on? The NK cells. But NK cell doesn't come in acquired immunity. NK cell we studied, that was innate immunity. Now we're talking about acquired immunity. So here we have B and T cells, and the question that comes is which of them is more predominant? So T cells are more predominant. Where are both of them formed? Of course, the formation of everything in my body has to occur in the bone marrow. So repeating once again, the formation of both the B and the T cells is going to occur in the bone marrow. But where do they mature? So remember, B cells mature in the bone marrow only, little bit in the lymph node, but predominantly in the bone marrow. That is why you call them B, na? They mature also in the bone marrow. They have birth also in the bone marrow. They mature also in the bone marrow. But T cells, why do you call them T? They have taken birth in bone marrow, but I'm still calling them T cells because they are going to go to T for thymus. So simple. Now I've understood the logic of calling them B and T. B because it was, where is it maturing? So for example, all of you have a birthplace. In दोनों का birthplace तो same था. Like you and your siblings, you have the same house, the same birthplace, but then both of you go to different colleges to study and to mature. So out of B and T, they were born in the same house, that is bone marrow. B cell decided that I am going to sit at home only and study. I'm going to sit and over there only and get mature. T cell decided that no, I will go somewhere outside my home and study. That is, it has gone to the thymus. Howsoever you may study, ultimately, where are you going to settle in life? Where will you and your brother or your sister settle? Your settlement will be in different cities, different houses. So remember, the sites are going to be these three. B and T cells, imagine they are staying in the same city. They stay in the same lymph node but in different houses. They stay in the same spleen but in different houses. They stay in the same GI tract but in different houses. So city same hoga, but they're going to stay in different houses. So for that, let me first make you analyze the structure of a lymph node. Quickly, we are going to draw the basic structure of a lymph node that I want to tell you. Not going into the basic structure which you will study in lymphomas, that is different. I'm just telling you a very basic outline. When I talk about a lymph node, the outermost area that you have is said to be the cortex. That you're going to have, that outermost area is going to be the cortex. Then you have an area around it, and that area around it obviously then becomes the paracortex. And finally, you have something in the center. So the area in the center is known as the medulla. So you have a cortex and a paracortex and a medulla. Pehle to do M for M. Medulla is going to have macrophages. Story is only simple. M for M. Medulla is going to have macrophages. What is in the cortex? Cortex is going to have B cells and paracortex is going to have T cells. How am I going to learn this without getting confused? Which is bahar? Which is bahar? Which is outside? So bahar or outside? Bahar means outside, na? So which is outside? Cortex is outside. So bahar, B cells are bahar, cortex. Now I can fill this up. Where will you find the B cells? The B cells are seen bahar. They are going to be seen in the cortex. Whereas the T cells are going to be seen inside. That is the paracortex. The first aspect is okay with everyone. B cells are bahar in the cortex, T cells are in the paracortex. Very good. Lymph node story is done. Now let's move on to the spleen. What all parts do you have in a spleen? Or rather than parts, let me ask you, what all pulps do you have in a spleen? That is a better question. Na? There is something known as a white pulp and a red pulp. Now this, so everyone knows that red pulp is red because of lot of blood vessels, sinusoids, blood vessels, red blood cell, that gives them a lot of red color. But what is it about white pulp? About white pulp, this is white because it has a lot of white blood cells, pale areas, white blood cells. Hai. So now tell me, where will the B lymphocytes and the T lymphocytes be present? Both of them have to be present in their white blood cells. They have to be present in the white pulp. They can't stay in the same house. I told you same city is okay, but they have different houses. So B cell is also in the white pulp, T cell is also in the white pulp, but T cell in a special location called PALS. Go back to your first year. What was PALS? PALS was periarteriolar lymphoid sheath. It was periarteriolar lymphoid sheath. That is what was PALS. So please note, we can draw one arteriole if this is a blood vessel or an arteriole. Periarteriolar would mean that the cells are around the arteriole. They are periarteriolar lymphoid sheath. That is where the T cells are present, but let me tell you, they are present in the white pulp only. B and T both are present in the white pulp, but T is present around the arterioles known as periarteriolar lymphoid sheath. So please write over here. In the spleen, both of them are going to be in the white pulp, but something special about the T cells is that in the white pulp, they are going to be present in the location called PALS. Last one, where are they present in the GIT? As soon as I say lymphoid organs in the GIT, first GIT you will start making villi, because as soon as I say GIT, students start making villi. Villi is nothing but going to have the epithelium. So please remember, one set of lymphocytes will be in the epithelium. One set of lymphocytes will be in the epithelium. So I will call them intraepithelial lymphocytes. I will call them intraepithelial lymphocytes because they are present in the epithelium portion, intraepithelial lymphocytes. You know, these are the T cells. These are the T cells. What is the other one? The other one is going to be present over here. Where is this? What are these big patches known as? Especially you see a lot of these patches in the ileum. कौन सा patches होता है? These are known as the Peyer's patches. Basically, this is the malt tissue. This is the malt tissue, that is mucosa associated lymphoid tissue. Mucosa associated lymphoid tissue, which in very simple words is known as Peyer's patches. So remember, malt, mucosa associated lymphoid tissue known as Peyer's patches. They are made up of what? They are actually made up of the B cells. So repeating once again, the cells which are in the epithelium, the intraepithelial, the tile, that is T cells are intraepithelial, whereas Peyer's looks like B, na? So Peyer's patches are going to have B cells, and finally your table is sorted. Tell me, in the GIT, intraepithelial lymphocytes are going to be T cells, whereas malt or Peyer's patches, whatever you may want to call it, that is going to be the B cell. Is your final table done for us? A quick revision. What is more prominent in the blood? T cells are more prominent. Where are both of them forming? Both of them are forming in bone marrow. Where do they mature? Bone marrow se nikal ke B cells to stay in bone marrow only. B for B. T cells go into the thymus, right? Talking about their sites, lymph node. B cells are bahar in the cortex. T cells are in the paracortex. In the spleen, both of them are in the white pulp, but T cells are present in pals. In the GIT, both of them are in GIT, but T cells are present in tile, that is intraepithelial lymphocytes, and B cells are B and P, that is in the Peyer's patches. Done with the cellular immunity of B and T commonalities, not done with their structure. Now we are going to take them up individually. We are going to study T and B cells individually, and I have drawn a T cell over here for all of you to identify. Now when we look at this T cell, every T cell has something called a T cell receptor. Every T cell has something called a T cell receptor. That is a receptor that I am going to draw over here like this. This receptor is made up of what chains? It is made up of alpha and beta. Remember, T cell receptor, in 95% of the cases, it is an alpha beta receptor. Only in 5% of the cases will it be the gamma delta receptor. But 95% of the cases, it is alpha beta. When it is gamma delta, by chance, that has been a recent previous year question. That has been a recent previous year question, that gamma delta T cells, which are hardly just 5, 7, 10% maximum, 95% is alpha beta only, gamma delta only 5%. They are going to be present in GIT, GUT. What do I mean by GIT, GUT? GIT gastrointestinal tract, GUT genitourinary tract. So, jo gamma, delta, gamma, delta, so GT, GT, gamma, delta, they are going to be GIT, GUT mein present hai, and they also do not require any MHC. They will process the antigen. What antigen? You can give them a lipid antigen, you can give them a protein antigen, but they will pick it up themselves. They don't need any MHC to come and serve it to them. No. So simple. What are the points for gamma delta T cells? They are very, very rare, 5-7% only. They are going to take up any kind of antigen, but they don't need anyone to give them the antigen. No MHC needed. And where are they present? Gamma delta. Repeating once again, where are they present? The gamma delta T cells are present in GIT, GUT, GIT, GUT. Okay? Coming back, the most common is going to be 95% cases having alpha beta T cell receptor. Next, moving on to the CD markers. When I'm talking about the CD markers, we have 1, 2, 3, 4, 5, 6, 7, 8, then 28, then 40. तो कैसे याद करें? अभी I'll tell you gradually, but for the time being, you guys have to learn 1 to 8, 28. Repeating once again, how will you learn it? 1 to 8, 28 becomes easier to learn. 1 to 8, 28. So this means all the CD markers that I'm talking about that are going to be present, they are all over here, and this one I want to give preference. Out of 1 to 8 and 28, CD3 I'm giving preference. Why? Because if someone asks you which is present on all the T cells, the pan T cell marker, the pan T cell marker is going to be CD3. And if someone says that, okay, antigen to aa gaya, antigen has come, but who will take the signal forward? Who will tell the T cell that, listen, your antigen has come, you start working? Who is going to do the signal transfer for us? That is also CD3. Please remember, CD3 is going to be not only the one which is a pan T cell marker, but also the one that takes the signal forward. Very, very important question. Next, which of these is known as the helper T cell? Helper T cell, I think everyone knows, known as CD4, so I write it as TH, helper T cell CD4. Cytotoxic T cells are going to be CD8. They do the killing. Cytotoxic, they do the killing, right? So helper T cell is CD4, cytotoxic T cell is going to be CD8. What is their ratio? Very easily students learn like this. CD4 to CD8 ratio is 2:1, but when they go in the exam, it becomes 1:2, 2:1, big time confusion. Remember, in the entire immunology, there is going to be a rule of 8 that we should study. Whenever, there will be a lot of places where you will have confusion. So whenever in confusion in mathematics, go for rule of 8. Means if you say CD4 upon CD8, was it 1:2 or was it 2:1? So I am saying it is 2:1. How do I learn it? Because I know that if I do a multiplication of 4 into 2, that will give me an 8. And if I do a multiplication of 8 into 1, that will also give me an 8. So either ways, I should get an 8. So 4 2s are 8, 8 1s are 8. So this seems right. If it would have been 1:2, to nahi hota, then it wouldn't have come out as 8. Na? So remember, when in confusion, you have to fulfill the rule of 8. Not only that, guys, in which condition does the CD4 upon CD8 ratio decrease? Whenever I say ratio is decreasing, matlab what is decreasing? Its numerator is decreasing. Which is that virus which attacks our CD4 T cells? It is always HIV. In HIV, don't you do CD4 counting because the CD4 cells are destroyed? Also, which is the situation where CD4 upon CD8 ratio increases, means the CD4 count increases. That is important. That is known as sarcoidosis. That is referred to as sarcoidosis. So repeating once again, when I say CD4 to CD8 ratio is decreased, means the numerator is decreased. The numerator is decreased, matlab HIV decreases the numerator. And when I say CD4 to CD8 ratio is increased, so CD4 increases where? It increases in sarcoidosis. Two very, very important questions in medicine. You have to mug it up, and that finishes off this slide also for you. Quick revision. When I talk about the T cell, which is the most common receptor? Alpha beta. How do you learn the CD markers? 1 to 8 and 28. 1 to 8, 28. Out of them, which is the CD marker which is most important? Pan T cell marker, signal marker, all of it is going to be CD3. What are helper T cells? CD4, cytotoxic T cells, CD8. What is the normal ratio? 2:1. However, if I ask you, who out of these is said to be the master regulator of the immune system? Master regulator of the immune system, and that is going to be CD4. The helping one is the master. The helping one is the master regulator. The master regulator, the helper, is going to be CD4. Master regulator, mastermind of the immune system. So this was about your what? This was about primarily your T cells. Now the T cells have some relation with the cancers. You know what? Let me tell you. So you've studied this in your chapter of neoplasia also. I'm repeating it once again. You know what happens? T cells have something known as PD receptor. And cancer cells say that, okay, what I'll do, I will now coat myself with PD-L1. That is what the cancer cells have done. So simple. If this is a cancer cell, for example, the cancer cell is going to coat itself with PD-L1 because the T cells have a PD receptor. And you know what is going to happen? When the PD-L1 and the PD receptor will bind, there is an immunosuppression that happens. Whenever the PD receptor is activated, there is a suppression of the immunity that happens. This basically results in an immunosuppressive response. So have you seen? Cancers are really smart. What are cancers saying? Cancers are saying that I know that by chance, every T cell has a PD receptor. If by chance I can activate that, the immunity of the person will go down and the person will not be able to kill me. So cancer ko ye pata hai. Cancer has got its ligand, your PD-L1 ligand, so that this pathway can be activated. Your immunity goes down and you can't kill him. But who is smart? Cancer or me? Of course, how can we get defeated by cancers? We are smart. So we decided, okay, cancer, no matter what you do, हमने अपना receptor ही block कर लेना. I am going to have some drugs called anti-PD-1 drugs. We are going to have anti-PD-1 drugs, for example, pembrolizumab, and that is going to block the PD. Then if this pathway is only not activated, how will you suppress my immunity? So end result, who is smarter, cancer or me? Of course, I am smarter. Okay, so repeating this once again, when I'm talking about the T cells, remember T cells have something called a PD receptor, and this cancer cell knows knows that if I will activate the PD receptor, the immunity of the person is going to go down. There is going to be immunosuppression. So what does cancer cell do? Cancer cell says, okay, I am going to get PD ligand, PDL1 with me, and when both of us will bind, then there will be immunosuppression. But then I decided that no, I'm not that dumb. I am going to give a drug and I'm going to block this receptor. If I block this receptor, how will you come and kill me? So that is known as pembrolizumab. I hope this cancer relation is also done. The T cells are over. Can I move on to the B cells now? Here I've drawn a B cell and you've got the same set of things written in front of you, but now you're going to compare it with T cell again and again. So let's get going. When I'm talking about a B cell or a B cell, the first thing that we have is a B cell receptor. B cell receptor. And do you know what a B cell receptor is? MD. Super lucky. I always say B cell, God has blessed. It has been blessed with MD degree. The B cell receptor is already by birth, it is IgM and it is IgD. Very jealous of B cell, isn't it? Okay, so the B cell receptor has MD degree already. Now, what are the markers that B cell is going to have? What are all the CD markers that it's going to have? Very difficult to learn, as you can see. Kuch is present in 10s, kuch is in 20s, some things are in 40s, some things are in 79. How will I learn so much? Let me tell you. I just told you the way. Some things are in 10s. Some things are in 20s, double ho gaya, then double 40 and then almost closing to 80. So double. B cell mein double. So 10s, 20s, 40s, 80s. So in 10s we have CD10. Repeating once again, in 10s we have CD10. Do you know what is the other name of CD10? CD10 is also known as CALA. What is CALA? So split it. C for common and ALA for ALL antigen. So C for common and ALL antigen. So CD10 is also known as CALA. Then 10s ke baad you are going to go to the 20s list. So this is what you have in the 20s. You have 19, 20, 21, 22, 23. Okay, out of these, remember 20s ka series is done na, then you have some pan B cell which I'll come to. 20s ka double kara, what do you go? CD40, simple. 40 ka double 80. So instead of 80, I've got two 79s with me, 79A, 79B. Repeating, you've got a 10, so CD10 is CALA. In 20s, you've got 19, 20, 21, 22, 23. Then 40 kara double, then 80 mein 79A, 79B. So now you will ask me, which of these is the pan B cell marker and which of these is the signal carrying marker out of these? Now guys, what was for T cell? Whether in T cell, I asked you the pan T cell marker or I asked you the signal marker, the answer for everything was CD3, but B cell mein answer is not common. The pan B cell marker is CD19, but signal marker is CD79A and B. How will I learn this? The pan B cell marker is CD19. But if I have to carry the signal, I've drawn two arrows. If I have to carry the signal through, how is it looking like? It's looking like a 79. Repeating, pan B cell marker is a 19. But if I have to carry the signal through and through, that through and through signal is going to be 79. Now you know pan B cell is 19, signal is 79A, 79B. Next question, recent exam question. How does Epstein-Barr virus enter the B cells? Because again and again I tell you, EBV ka B yaad rakho. Epstein-Barr virus enters the B cells. How does it enter the B cells? So if this is an Epstein-Barr virus and it has to go and attach to the B cell, it will search for CD21CR2. That's the full name, CD21CR2. So repeating, out of the 10s and 20s and 40s and 79, remember 10 is known as CALA. 19 is the pan B cell marker, but signal transduction is by 79A, 79B, and the EBV entry is by CD21. That wraps up your B cells. But if you have noticed, I have spoken about B cells separately, T cells separately. Why am I not talking about them together? Because at the end of the day, they are cousin brothers. They will talk to each other. How is the interaction of B and T going to be? And I'm also going to give you a life lesson. That life lesson is what? That life lesson is that be happy with what you have, because B cell is not happy with what it has. Can you recall for me, if this is a B cell and then this is a T cell, can you recall for me, B cell was blessed with what? B cell was blessed with 2 antibodies. कौन सा degree already blessed है? B cell is blessed with MD degree. But no one is happy with what God has given them. B cell has MD degree. B cell says, no, I'm not happy with MD degree. मुझे super specialization करना. I want to do something more in life. So initially we are having IgM and IgD, but then we say that no, I want all the antibodies. I want the entire spectrum, just not IgM and D. I want the entire GAMD, IgG, A, M, D, E. I want everything. What is that known as? That is known as isotype switching. That is referred to as isotype switching. So repeating once again, you are going to convert MD degree into all of that. That is known as isotype switching. So please note guys over here, how will that isotype switching occur? So instead of switching, I have called it change. You know why have I called it change? Because I will first have to tell you a mnemonic. The way you write change, you write 40. 40 is what? 40 is going to be 40. And you write char. Char is what? Char is 4. So there is some 40 and some 4 that is going to come. So remember, for the change, I need char 40 ki last local. You remember that movie? Char 40 is what I need. For change, I need char and 40. So please remember, CD40 has been given something, B cell has been given something called CD40 receptor, and T cell has been given something referred to as CD40 ligand. Now, most of the people in the exam forget who had the receptor and who had the ligand. So let me clarify that also for you. The way you write a B cell is the same way you write R. B and R same likha, so B cell has the CD40 receptor, and the way you write T cell, T looks almost like L, so that is going to have the CD40 ligand. So first you have analyzed it. Then your next query will be, what if in the exam only CD40 is written? What will I assume that to be, ma'am? I will assume that to be the receptor or the ligand. I will always assume that to be the receptor. Remember, CD40 alone written is always considered as receptor. So if they write CD40 or they write CD40 receptor, that is the same. When they want to tell you the ligand, they will write the word ligand over there. Okay. Now, moving forward, I realize that whenever an antigen comes and there is a yellow color bacteria that has come, this B cell says that akele to I can't do anything. Let me give this bacteria to my cousin brother T cell because I am not happy with MD degree. Mujhe to super specialization karna. I want GAMD degree. So let me ask for help from my cousin brother. It picks up this antigen, gives this antigen to the T cell. The T cell very innocently makes which interleukin? Char, interleukin 4. And interleukin 4 is then going to go and change that MD degree into GAMD degree. So remember, why did I say change requires char and 40? Because they will ask you which interleukin is needed for isotype switching. Now you know which interleukin is needed for isotype switching. It is interleukin 4. And which CD marker is needed for isotype switching? It is 40. So for the change, I need char and 40, interleukin 4 and CD40. I hope I can repeat it once again in 15 seconds. B cell was born with MD degree, but it's no one's happy with MD degree. So B cell wants GAMD degree. So that is known as isotype switching, which is the CD marker that is going to help it. CD40 is going to help it. And which is the interleukin that shall help it? That is interleukin 4 will help it. So ultimately char, 40, and that is going to help in the change and result in GAMD degree. So guys, till now I was only talking about BT cells, cellular immunity. Finally, I have made all the antibodies. So which immunity have I entered into? I have entered into humoral immunity. What is humoral immunity? Humoral immunity is the formation of antibodies. And why do I always write it as GAMD? I always write it as GAMD because I'm going from the maximum concentration all the way down till the least concentration. From the maximum concentration to the least concentration. And this is very important. They will ask you which antibody has the maximum concentration in the blood. This was a recent FMG exam question. And which is the antibody which is going to have the least concentration in the blood? That is Ig. So always remember GAMDE. GAMD is the decreasing order that we have. I'm not picking up every antibody in detail because that's a separate chapter that we have studied in the microbiology section. So I hope all of you can easily pick it up from there. But for the time being, yes, you know that your humoral immunity is sorted. But now the problem is that I have finished all these antibodies and I've finished all these 2 types of immunities. Innate is done, acquired is done, but I've studied it in a scattered manner. Let me put a combination. The body doesn't work as B alag and T alag and everything alag alag. No, body doesn't work separate. Body works as a a team, as a good group work, group effort is put up. So let's talk about a story. Like today, if a bacteria enters into my body, what will be step number 1, step number 2? How are the cells going to get activated? That is the workflow that I'll be talking about next. Having discussed the normal cellular and humoral immunity, now let's go on with how these cells would work. So what is the activation process going to be? I'm going to draw one entire flowchart, and at every level I will keep explaining what is happening. So first and foremost, let me draw one barrier of the body, and let that be the skin, for example. So let me draw some tiny little hair over here. So just as an example, there is a bacteria. This is a bacteria which has tried to enter via the skin. So bacteria I'll consider as antigen, right? So the bacteria will try to enter via the skin, and which is the first cell which is going to pick it up? The first cell that shall pick it up is an APC, that is antigen presenting cell, the first cell to pick it up. So I'll say that the yellow color antigen has been picked up by the antigen presenting cell. So your next question is, how many types of antigen presenting cells are there? So I have an entire flowchart, but at every step I'm going to explain things to you. So how many types of APCs? As I said, we are talking about APCs, antigen presenting cells. There are two types of antigen presenting cells called professional APCs and non-professional APCs. When I say professional APCs, we have something called DBM. What is DBM? That is dendritic cells. The dendritic cells are there. Next we have the B cells, and lastly we have the macrophages. So repeating once again, the dendritic cells, the B cells and the macrophages, these are going to be the professional APCs. Professionals do most of the work. So they do most of the work, out of which please remember these dendritic cells are going to be present in two places. Firstly, the dendritic cells can be present in the skin, and over there I think everyone knows that we call them the very famous Langerhans cells. Langerhans cells, not Langhans cells. Why? Because Langhans cells were not any antigen presenting cells. Langhans cell was a giant cell that you see in TB, totally different. Whereas Langerhans cell is the antigen presenting cell in the skin. Then what is the antigen presenting cell that we have in the lymph node? There is going to be a follicular dendritic cell. That is known as follicular dendritic cell. And do you know what is fancy about this follicular dendritic cell? This is where HIV stays for years and years. You must have heard this about an HIV, that HIV virus comes in the body, but AIDS happens say 7 or 8 or 10 years later. So where is HIV sitting in that duration? What is the reservoir for HIV? The reservoir for HIV is going to be the follicular dendritic cells, present in whose follicles? Present in the lymph node follicles. Okay. So when I talk about dendritic cells of two types, one is in the skin known as the Langerhans cell, and one is in the lymph node known as the follicular dendritic cell, and this is where the HIV is going to stay. These are the professional ones. What are the non-professional ones? We have thymic epithelial cells. We have thymic epithelial cells. Then we have endothelial cells. Endothelial cells, and finally we have fibroblasts. They are also important, not as important as the professional category, but thymic epithelial cells, after epi you will learn endothelial cells, and then you will have fibroblasts. These are the non-professional ones. So if I come back, I say that whenever an antigen will come, who will pick it up? Which is the first cell which is going to pick it up? The professional and the non-professional APCs will pick it up. But did you focus on its name? Antigen presenting. It has to present that antigen. The antigen is not going to be kept inside it. It has to present this antigen to someone else, and that someone else is a T cell. That someone else is a T cell. So it has to present the antigen. Just imagine, consider that this antigen is like a guest who has come into your house. Okay, so now when a guest comes into your house, if you have to give him water, will you just go and throw water in his hand like that? No, you will take a nice plate, tray, you will put the glass of water in it. Basic etiquettes, you have to make sure that the guest feels comfortable. So am I just going to throw this antigen like this, like a ball? Am I going to throw it onto the T cell? No, because I am going to put it in a nice little tray. What is that nice little tray known as? Antigen presenting cells have MHC. So I'm going to first put this antigen on an MHC tray, and then I am going to give it to the T cells. So your next question that you have to study with me is, what is MHC? So देखो APC हो गया. Now next what I'm going to teach you is MHC. First you should know the full form of MHC. What is MHC? MHC stands for Major Histocompatibility Complex. It is going to be many types. You will have MHC1, MHC2. But before I tell you all of that, how did MHC come from? From which gene does it come from? It comes from HLA gene, which is present on chromosome 6p. 6p means the short arm of chromosome 6. It is present on the short arm of chromosome 6. Chromosome 6p has the HLA gene, and that gene will give rise to this MHC molecule. Before I proceed, can you tell me all the things in pathology, or even in, for that matter, micro or medicine, all the things that you study for chromosome number 6 that are important, even important in path, in medicine, and micro, everywhere. So first and foremost, the HLA gene that is present on chromosome 6. There is one more gene present on chromosome 6, and that is going to be the HFE gene. HFE gene is the gene for what? Fe. Think of iron overload. HFE gene is the gene for hemochromatosis. That is also present on chromosome 6. And remember one more thing in pediatrics and medicine. Autosomal recessive polycystic kidney disease, that is also going to be on chromosome 6. So repeating once again, chromosome 6 has the HLA gene, it has the HFE gene, and it has autosomal recessive polycystic kidney disease. All of these on chromosome 6. So coming back to the HLA gene, do you want me to draw one HLA gene? Life is going to get a little more simpler. So when I have this as, and this is not one. So see, what have I done? Everyone draw with me. This is one HLA gene, but it has many parts. And what are those parts going to be? I am going to divide it like this. Means this is HLA gene 1, this is HLA gene part 2, and this is HLA gene part 3. In the order of their discovery, it is 1, 3 and 2. Okay? Further, I will divide them. There is HLA A, B, C. After C, what's the next alphabet? D. D I take to the other end. D, D, D means what is D, D, D? It is DP, DQ, DR. DP, DQ, DR. So remember, A, B, C is in the first aspect. Then D, 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 DP, DQ, DR is in the second part of the gene. Then where are the other ones? What about E and G? You come back. E and G are again back in HLA gene 1. So 1 and 2 only have parts. This third one I'm not going to make any parts out of. Only in 1 and 2. What do I mean by that? HLA 1 has ABC. Then D goes all the way in the other part, and then E and G comes back. Now, before I start off with the story of MHC from here, let me tell you the things that you have already studied. I want to mention something about HLA-E and HLA-G, which is important. If you guys remember, long back in the beginning of this chapter, we did something called natural killer cells. I'll quickly remind you of that story. Natural killer cell had an arm called the inhibitory arm, which said that whenever you have any self-antigen and whenever you have any self-cell, that self-cell will always express MHC1. So, agar koi apna ID card you saw, remember the ID card story? If you see your own ID card, you will always put out your inhibitory arm and not kill that cell. You will know that, okay, this cell is my cell. I'm not supposed to kill it. For your own cell, you will never ever kill it. So how did you get to know what is the ID card? The ID card was MHC1, and now you know MHC1 ka kaunsa wala part, HLA MHC1 ka kaunsa part. So remember, it is the HLA-E which is going to be the self-recognition target. Remember, for the NK cells, the recognition target is which part of MHC1? It is actually HLA-E gene which is that target. Now you know what I'm talking about. So sab kuch I, E. So for I myself, the ID card was HLA1. HLA1 ka matlab which part? It is going to be E, again sounding like I. And which arm of NK cell will come forward? Inhibitory arm will come forward. Next, we also have something called HLA-G, G for gestationalis. G is going to be for gestationalis because this is responsible for gestation, feto-maternal tolerance. So, have you ever wondered that if a tiny little bacteria also comes into our body, our immune system starts working 24/7? A tiny bacteria, tiny virus. Now, just imagine that in a mother, when an entire human being is growing, in a pregnant lady, when an entire human being is growing, that is a fetus, why isn't the mother attacking and treating the fetus as a foreign element? Because the fetus has coated the self with HLA-G. This small little fetus, he has coated himself with HLA-G. So as soon as the mother will see, oh, this is HLA-G, this is my own gestation, I do not have to treat it like any foreign body. This is my own bacha, right? So remember, HLA-G is responsible for feto-maternal tolerance, and HLA-E is responsible for the NK cell target. This is the extra point that you had to know. Once you've done the structure, now I can tell you that HLA-1 gene gives rise to MHC1. Wow, I have not told you any rocket science, it's common sense. HLA-1 gene will give rise to MHC1 protein. Similarly, another rocket science question that I'll tell you is HLA-2 gene is going to give rise to MHC2 gene. How nice. HLA-1 gives rise to MHC1, HLA-2 gives rise to MHC2. What does HLA-3 give rise to? So there is nothing known as MHC3. That's the first thing. There is nothing known as MHC3. So HLA-3 gives rise to complement proteins like C2 and C4. It can give rise to the complement proteins properdin. It can give rise to TNF-alpha. It can give rise to heat shock proteins. It does not give rise to any MHC. So take home message, MHC, major histocompatibility complex. Either it is of type 1 or it is of type 2, which will come from HLA-1 and HLA-2 respectively. There is nothing known as MHC3. HLA gene 3 hai, but no MHC3 gonna come out of it. For HLA-3, we have complement system ke things like C2, C4, properdin. Then we have TNF-alpha and heat shock protein coming from it. So now I know that I just have to do the differences between MHC1 and MHC2. Others don't exist only. So MHC1 and MHC2, what is the difference? So first and foremost, I'll draw a structure. So everyone can draw this with me because it will really help them, in learning the process. So I have drawn cells like this. What does that mean? And in the other one, I am going to draw something like this. When I'm talking about MHC1, you have to write in a clockwise manner. Matlab, the first chain is the alpha 1 chain, the second chain is the alpha 2 chain, the third chain is the alpha 3 chain. Have I written it in alphabetical order? Alpha 1, alpha 2, alpha 3. And what is this separate looking chain that I have in pink? This is the pretty pink beta 2 microglobulin. This is the beta 2 microglobulin. So it has two chains. Firstly, the alpha series, alpha 1, alpha 2 and alpha 3. And then it has one separate chain which is beta 2 microglobulin. So now, why am I reading this MHC? Is because I know for a fact that MHC was a tray and I had to keep an antigen, a glass of water. Where should I keep the antigen in this structure? That yellow color antigen is kept over here at the junction of what, guys? What is the antigen binding cleft? It is kept at the junction of alpha 1, alpha 2. Previous year question. It is kept at the junction of alpha 1, alpha 2. Now let me draw MHC2. How have you drawn MHC2? How do you write 2? You write 2 as a up, down, up, down. Writing it again, up, down, up, down. That is how you write 2, because the labeling will also be up, down, up, down. This is alpha 1. This is alpha 2 chain. This is beta 1 chain. This is beta 2 chain. So alpha 1, so simple, alpha 1, alpha 2, beta 1, beta 2. Where should I keep the antigen? Where have I kept the antigen? I have kept it as alpha 1, beta 1 is the antigen binding cleft. So in the exam, when you will go, you will get confused. Where was alpha 1, alpha 2? Where was alpha 1, beta 1? Major confusion hota hai. So very simple. MHC1 has only one, which is the first alphabet A. So isme all the A's have to be there. Alpha 1 and alpha 2. You can't keep any beta. In MHC1, all the A's have to be there. Whereas in MHC2, the way you write 2, first you write 1, matlab A alphabet, then you make it a 2, matlab B alphabet. So, in simple words, A will also be there, B will also be there, A will also be there, B will also be there. What is the antigen binding cleft? Alpha 1, beta 1. Repeating. For MHC1, all the A's have to be there, alpha 1, alpha 2. For MHC2, A and B both have to be there, alpha 1, beta 1. Finally, you will ask me that where are these present in our body? So MHC1 is going to be present on all the nucleated cells of the body. MHC1 is present on all the nucleated cells of the body. It is also present on the platelets. So now tell me, does red blood cell have a nucleus? No, na. So red blood cell is not going to have it. Also, please remember, sperms don't have MHC1. But kuch problem yaad aa raha hai? I just told you that everyone who has MHC1, that was your ID card. If you guys remember very carefully, MHC1 was your ID card. That It will tell you that, okay, this is my own cell. So RBCs are not, they have misplaced their ID card. Now, how will I get to know that these are my own cells? So RBC says what? So now RBC is trying to act very smart. RBC says, I to have lost my ID card. I don't have MHC1, but survive to karna hai. I have to go in class. I have to attend the lectures. So how will I do that? So RBC says, it's okay. Let me take someone else's. One of my friend is sleeping in the room. Let me take his ID card. Who will get to know? So what does RBC do? RBC basically tends to, very, very important thing that they will ask you in the paper, that RBCs do not have MHC1. So how do they protect themselves? They are going to absorb MHC1 from the surroundings. It is going to absorb MHC1 from the surroundings. Very, very, very important point that you have over here. So repeating once again, you might feel RBC doesn't have ID card, we are going to kill it. No, we don't, because RBC is smart. It has taken his friend's ID card. He has absorbed MHC1 from the surrounding and made himself enter the class, made himself secure. So coming back, MHC1 is present on all the nucleated cells and the platelets. It is not present on red blood cells, not present on sperms. Where are MHC2 present? MHC2 is present on all the APCs. It is present on all the antigen presenting cells, right? APCs. So now I've done the structure of MHC. I have understood that, okay, MHC is present on these cells, but what was MHC doing? MHC was the tray, and the tray had to give water to a T cell. How many type of T cells do I have? We have CD4 T cells, we have CD8 T cells. So now you will ask me that which MHC is going to give to which one? MHC1 presents the water, presents the antigen to which cell? It presents it to CD8, whereas MHC2 presents it to CD4. How will I learn this aspect? Rule of, whenever confused in mathematics, what is the rule that you have to know? You have to follow the rule of 8. What is the rule of 8? That if you're dealing with MHC1 and you get confused कि अच्छा CD8 को या CD4 को, whom is it giving it to? So 1 8s are 8. This means MHC1 gives to CD8 and MHC2 is going to give to CD4. So 4 2s are 8. That is how you analyze the rule of 8. Where else did we study the rule of 8? Very, very important point that you have to know. Please note, if they ask you what is the CD4 upon CD8 ratio and you get confused, 1:2 or 2:1, we had studied complete the rule of 8. 4 2s are 8, 8 1s are 8. So it's going to be 2:1. Similarly, you get confused MHC1 versus MHC2. Which CD cell does it present to? So 1 will present to CD8. 2 will present to CD4. So this is going to be one of your rule of 8 that you follow in path, micromedicine, everywhere, immunology. And this is the other rule of 8 that you're going to follow. So that finishes off the entire story of the rules of 8. No other rule of 8 I'm going to tell you in the entire immuno section. Coming back, now let me also include biochem in this. Why only path and micro? So if I have to do the testing for MHC1 and for MHC2, what are the tests that I do? This is just some little bit of ratta that you have to do because it's important in biochemistry. So remember, for the diagnosis of MHC1, we have something known as allo-anti-sera testing. We have allo-anti-sera testing, and for the diagnosis of MHC2, we have MLR, mixed lymphocyte reaction testing. We have mixed lymphocyte reaction testing. Repeating once again, for MHC1 diagnosis, we are going to have allo-anti-sera. We will have allo-anti-sera testing in our lab, whereas for MHC2 testing, we are going to have MLR, that is mixed lymphocyte reaction. Thoda sa biochem, and that's about it. So coming all the way back, guys, so much so we have done in our flowchart. We have done antigen presenting cells. Antigen presenting cells have a tray, that is MHC. We've done everything about MHC. Now the MHC has either given it to CD4 or to CD8. So let us quickly study about these. CD4 were the helper T cell. CD8 were the cytotoxic T cells. Hai na? Do you want to study them in detail? Why not? Let's first study CD4 T cells. That is the helper T cells. And I hope you guys remember the CD4 or the helper T cells were defined as the master regulators of the immune system. They are known as master regulator of the immune system. Right? So they are going to control everything. So these T helper cells, which are going to control everything, are actually of 3 types. That is TH1, TH2 and TH17. TH1, 2 and 17. So first and foremost, what does TH1 release? TH1 release interleukin 2, It also releases, or not releases, but is controlled by interleukin 12. So it has an association with interleukin 12 and also interferon gamma. So I have learnt it as, what are the interleukins associated with TH1? तो 1 के साथ 2 है, तो 1 2 को जोड़ो, that is 12, that is the next number that you get, and interferon gamma. Please remember, with interleukin 2, it's like a vicious relationship. Means I will promote you, you will promote me. Simple as that. So if I teach you well, you will answer my questions well. Hai na? Symbiotic. Same way, TH1 releases interleukin 2. Interleukin 2 is going to activate TH1. That is very, very, very important that you have. So TH1, interleukin 2, it is also controlled under the effect of interleukin 12, and interferon gamma. I think everyone knows something about gamma. G for gamma, G for granuloma. This is the one that is going to help in granuloma formation. Repeating, G for gamma, G for granuloma. This helps in granuloma formation. All the TB and leprosy wala granulomas, they are going to be under the effect of interferon gamma. Coming to the next, that is TH2. TH2 releases everything that you have in asthma. What all do you have in asthma? You have interleukin 4, interleukin 5 and interleukin 13. 4, 5 and 13. Did I just teach you what does interleukin 4 char? Remember char chalis, what does it do? It helps in isotype switching. It makes all the other antibodies. It helps in isotype switching. Interleukin 5. So char chalis is simple. 5, A, B, C, D, E. Which is the fifth alphabet? It is E. So it is going to result in eosinophil activation. E for fifth alphabet. So remember, eosinophil activation. And interleukin 13 is going to help in mucus production. Why did I say this is the entire asthma wala story? Because asthma allergies, hypersensitivity type 1, they show you antibodies, they show you lots of eosinophils, they show you a lot of mucus. Asthma shows you a lot of mucus, a lot of eosinophils and a lot of antibodies. So interleukin 4, 5 and 13 are going to be responsible for type 1 hypersensitivity. Coming to the last one, interleukin 17 will come from where? Common sense. TH17 will release interleukin 17 and 22, and they are mainly, you don't have to know much about it, but they attract the neutrophils and the monocytes. They are responsible for those. So repeating it once again, remember TH1 is going to be 1, 2, 12 interferon gamma. TH2 is 4, 5 and 13, and TH17 is 17, that is interleukin 17, and T helper cells are the masterminds. Who is the other one other than T helper cells? What is the other one that we have? We have the T cytotoxic cells. We have CD8 cytotoxic T cells. Their name, cyto means cell. Toxic means killing. They are going to kill the cell. So the question comes is, how do they kill the cell? What is the mechanism of killing that you have to know? Just 2 mechanisms which are sorted for you. One mechanism you already know. Do you remember CD95 and CD95 ligand wala system? What was CD95 and 95 ligand? Basically, apoptosis. So go back to your apoptosis chapter in pathology, general path section, and you will study that we have apoptosis where CD95 and 95 ligand help into play, and I'll draw it once again. This was the cell which was wanting to die. So this had CD95, and there came a T cell which you might thought much about over there because you were doing apoptosis. Now you did not think much about ke kaun sa cell, but that T cell, that CD8 T cell had something called CD95 ligand, and that is what was important for you to know, that T cells are going to help, CD8 T cells are going to help in apoptosis. So first mechanism is CD95 ligand apoptosis. Next is going to be a mechanism known as the perforin granzyme system. Next is known as the perforin granzyme pathway. So what is the perforin granzyme pathway? The name is quite nice. Perforin, I am going to create a perforation, and in that perforation, I will release my enzymes, granzymes. So what do I mean by that? For example, if this is a CD8 T cell, this has 2 things. It has perforin and it has granzyme. The CD8 T cell has perforin and granzyme, and it comes across some bacteria, and that bacteria has to be killed. How will it kill it? First, it will make perforations. First, the P is going to go. The P is going to go and make perforations. Who is going to enter through the perforations? The G, the granzyme, will then enter through the perforation and kill it. So simple na concept. If you know perforin and granzyme, तो clear हो जाएगा. Let's do a quick recap. So for example, if this is a bacteria, the first thing that I'm going to create is perforations inside it. I will create perforations inside this bacteria. Via that perforation, what will I send? Via that perforation, I'm going to send the granzyme. I will be sending the granzyme via that perforation so that I can manage to kill this entire bacteria. This is known as the perforation, the granzyme enzyme. So perforin granzyme pathway. Simple. CD8 T cells are going to help in killing either by the apoptosis method or by the perforin granzyme method. So coming all the way back to my flow chart, you had two cells, either CD4 or CD8. CD4 cells were either T helper type 1, T helper type 2 or T helper 17. Whereas CD8 cytotoxic T cells, they could either do killing by CD95 and CD95 ligand pathway, or they could do killing by the perforin granzyme pathway. Have we finished all the T cells? Yes. But you have also finished the other one. Were T cells working in isolation? Or do all the T cells constantly talk with the B cells and isotype switching occurs and all antibodies are formed? Gamd wala story comes into play. Did we study this? Repeating, all these T cells are constantly going to talk to their brother, that is B cells, and B cells are then going to make what? B cells are going to make all the antibodies, that is Gamd story is going to form. Hai na? Now you can say the entire pathway is clear. Whenever an antigen comes, the first cell that picks it up is the antigen presenting cell that is going to be served on a tray called MHC to a T cell. The T cell is going to be either CD4 or CD8. CD4 helper T cell is of three types, Th1, Th2, Th17. CD8 T cells are also going to kill by two mechanisms, the apoptosis or the perforin granzyme system. The T cells are always going to interact with the B cells, and the B cells are going to release all the antibodies called Gamd. I hope this entire activation mechanism is okay with everyone, because we are ready to solve one quick question and wrap up this topic. Which of the following true statements about MHC are all except? These are the questions that you have to read very carefully. That except word, is a problem. Tell me a true and false in this, okay? Does chromosome 6 harbor the gene for MHC? HLA gene, is it present on chromosome 6? That seems to be true. Next. Gene encoding for complements are adjacent to MHC. What is this? So if I remember my HLA, it was 1, 2 and 3. 1, 3 and 2 is how we used to write it. So HLA 1 and 2 used to make MHC 1 and 2. They used to make MHC 1 and 2. And what was HLA gene 3 making? HLA gene 3 was making all the complement proteins. So is the gene encoding for complement adjacent to the MHC? Yes, true. Next, look at this. Monocytes have class 2 MHC on their surfaces. So where were the MHC1 was all on the nucleated cells of our body and also on the platelets of our body. MHC2 was all the antigen presenting cells. What were the antigen presenting cells? DBM. Antigen presenting cells were dendritic cells, B cells, macrophages and monocytes belong to the same lineage. So this is also a true statement. Do the monocytes and macrophages have class 2 MHC molecules on their surface? Yes. Coming to the last one. Class 3 MHC पहले तो exist ही नहीं करता, right? There is something called HLA gene 3, definitely. HLA gene 3 exists, but class 3 MHC does not exist only. So, I have anyway got my false statement. Answer is there in front of you. So, this is the answer that they were asking you for, with which you have finished off your activation mechanisms.